Hi everyone, my name is Marco Savarese from Helsinki, Finland, and I wish first of all to thank the organizer for inviting me and to give me the opportunity of sharing our research with you all. Until 10 years ago, human genetics was based on a strong paradigm, the so-called one gene, one disease. We all thought that variants in a single gene can cause a single, specific, and well-defined clinical presentation. At the same time, we thought that each defined clinical condition was due to a small change, a small variant in a single gene. In the last 10 years, we have been able to analyze thousands, millions of different DNAs, checking one by one each specific nucleotide, each specific letter. And one of the main findings of this huge global effort is that we all realize that the old paradigm, one gene, one disease, is wrong. Medical and clinical genetics is much more complex, and different variants in the same gene can cause multiple different clinical conditions. This is particularly true for titin. We all know that small change in titin, small variants in the gene, cause a broad spectrum of muscle diseases, affecting only the heart, for example, resulting in a dilated cardiomyopathy, or causing skeletal muscle disorders, myopathies with or without a cardiac involvement. In particular, we know that only specific titin variants, only specific changes in titin gene increase the risk of developing a dilated cardiomyopathy. But we also know that often the cardiomyopathy develops in elderly individuals, meaning that additional factors, genetic and non-genetic factors, so including genes, but also non-genetic, for example, environmental factors, may increase the risk. And thereby, these additional factors are crucial to really develop a cardiomyopathy, also in presence of specific titin variants. Similarly, we know that titin-related myopathies can have different onset, meaning that the first symptoms may occur during the fetal development or in the first days or weeks or months after birth, but also later, eventually in early adulthood or later in the life. And at the same time, symptoms, their severity, and how the disease progresses may also differ. Why? Because titin is a very, very big gene, and its size explains what we right now call phenotypic variability. Its size explains the different clinical presentation linked to titin variants. And it's crucial to really study this variability. Is it really crucial to understand specific variants and their related clinical presentation? How do we do that? How do we study this variability? Well, first of all, we try to study titin gene in a large, very large number of individuals, with or without any clinical presentation. But we also need to carefully improve our understanding of how titin protein works. Let me give you a quick example of this. Titin is a large gene. Think of a very big book with several chapters containing instructions on how to produce the protein. We call these different chapters, these small parts containing instructions, exons. Well, for titin, not all these exons are really used in muscles. not all these chapters are really translated, used to make a protein in adult, in postnatal muscles. Some of the chapters are somehow skipped in postnatal muscle, although they contain crucial information for a correct formation of titin protein in fetal muscle, in muscle before birth. Why this is so important? Because a single variant, a single letter that is changed in these specific exons, in these specific chapters, which are not really used, that are skipped in postnatal muscle, these small changes definitively cause or result in a clinical presentation, a form of congenital titinopathy, with first signs during fetal development. And this is a very well recognized titin-related clinical condition. This was just an example, but why is it so crucial to understand how the different titin variants may result in different medical conditions? Well, first of all, to improve the diagnostic part, to really understand the clinical meaning of the small variants we find in titin. Then, to eventually discuss with all the individuals with titin-related medical conditions, how the clinical condition may evolve, and eventually, as we all hope, to produce as soon as possible treatments, possible therapies. And actually, Professor Ruud in Helsinki coordinates an EU-funded project aiming at improving our current knowledge of titin and titinopathies. And the project includes clinicians, scientists with a different background to study different aspects of titin as a gene, but also as a protein, which is crucial then for muscle contraction, in order to understand the molecular cause of titin-related, of the wide spectrum, I will say, of titin-related medical conditions. And a tangible result of our effort within this, what we call HGPRD, this EU-funded project, is visible in this poster that our brilliant student Victoria has presented. And her poster well summarized our study on one specific titin-related medical condition, the tibial muscle dystrophy. That is relatively common in Finland. So with this, I wish to thank all of you and Professor Ruud and Dr. Hackman for their huge work on titin in the last 25 or 30 years, and all the members of our groups in Helsinki. And please, if you have any questions, do not hesitate to contact me. Thanks again.